Today we are going to look at DNA packaging and the forces that contribute to it, as well as how the free energy equations we have learned in class relate to forces a bacteriophage undergoes during packaging. In bacteriophages, DNA is highly packed and forms a nearly crystalline structure. From the packaging density equation in class, we found that the packing density of a lambda phage is 0.6. As you can see in this animation of a 529 phage, the linear DNA is being packed into these concentric loops. Experiments show that as the capsid begins to fill, the packaging rate decreases as the force increases. This makes sense because at high packaging density, there is a large free energy cost to package the whole genome, which in turn leads to higher forces. In order to understand the relationship between free energy and force, we must first look at the free energy equations. There are two components that contribute to the overall energy involved. First, we have the energy involved in bending the DNA strands into the packaging shape, G-bend. This energy is a result of the resistive force involved when bending a strand of DNA that has stiffness. The second component contributing to the overall energy is that of DNA charge interactions, G-int. DNA subunits, when put close together, have repulsive and attractive forces that depend on the distance between each strand. When the distance between strands is greater than the preferred distance, there is an attractive force. When it is less than the preferred distance, there are repulsive forces. These two components are what contribute to the overall expression for energy involved in packaging DNA. G total is equal to G bend plus G int, which is shown in this equation. This equation gives us the total packing energy as the DNA length increases. To obtain the force exerted during DNA packaging, we need to differentiate that equation with respect to the DNA length L. By applying the chain rule to the left part of the expression, we get the following expression. For the right part of the expression, we can simply take down the coefficient for L and get the derivative. Now all we have to do is simplify the equation, and finally we obtain the expression for force. Now we can do some interesting analysis based on the equation for packaging force. Specifically, in this video we will look at how DNA packaging force is affected by salt concentrations. Looking at more of the parameters that affect the packaging force, we can focus in on how the maximum force scales with the packaging density. In this figure, we can see that the packaging force increases linearly with the packaging density and that this holds true across different types of bacteriophages. Concepts we have learned in Chapter 9 can also be applied to this situation. In Chapter 9, we focused on screening and how it can affect interactive forces between two anions. We learned that when charged macromolecules such as DNA are in ionic solutions, cations will surround it, minimizing the negative charge felt around the DNA. Applying this to DNA packaging into a capsid, this graph plots the maximum force against decreasing salt concentrations of the ambient solution. The repulsive interactions between the DNA strands grows progressively larger as the concentration of the sodium ions in solution decreases. This is a result in larger packaging forces. From our analysis, we found some interesting applications. First, we found there to be some possible medical applications. Understanding the forces involved can allow for the development of therapies that can disrupt the delicate balance inside the viral capsid. By altering salt concentrations, an increased capsid pressure could cause it to rupture, neutralizing the virus. Second, there is a possibility of virus-like therapies. An understanding of these forces could allow for the engineering and construction of virus-like medicine delivery mechanisms. Finally, understanding these forces can allow for a better understanding of DNA regulation and all the implications that follow from that. In conclusion, using structural biology, single-molecule biophysics, electron microscopy, and solution biochemistry, viral packaging could be addressed in a quantitative way. Looking at a paper by 2 of the 4 authors from our textbook, we took a more in-depth look at how ionic concentrations can affect packaging forces. Through this, we were able to apply concepts learned in previous chapters, such as Chapter 9 screening, applied to Chapter 10 packaging. The key insight we found from the force analysis was the dependence of DNA ejection inhibition on solution conditions. The repulsive interactions between the DNA strands grow larger as the concentration of sodium ions in solution decreases. This results in larger packaging forces.